system, you've all expressed an interest in our third line of defense. Now, that's fine and all, but this isn't for the faint of heart. Here's a picture of our staff on hand last week with the boss. Now, before we get started, I'll run through the uniform colours designated to each profession, or part of the immune system if you will. Now, these aren't all the parts of the immune system, however, they are all of the parts which I'll be introducing you to. Keep in mind, all of these components should be displaying the marker molecule which tells all of the components of the body that they are from this body, otherwise they would be classified as being non-self, which is what we call that here in the human body's immune system. Just now we saw the macrophage and lysosomes, which are part of the process in the second line of defense called phagocytosis. My name is Miss T, and I am part of a differentiated group of T-cells called the cytotoxic, or killer, T-cells, which you would have seen earlier. But enough about me, let's jump onto the second line of defense with phagocytosis. After that, I'll show you how that links to our role in the cell-mediated response team in the third line of defense. Righto, so when pathogens get past our second line of defense, they encounter a small, generally slow to respond, group of non-specific mechanisms, one of which being phagocytosis. All of these processes respond against all pathogens in order to minimize the spread of the pathogen in the boss's body. These pathogens are now referred to as antigens. Macrophages and neutrophils both come under the broader term of phagocytes, but for the time being, so that we don't get confused with the process and the mechanism, I'll just refer to the macrophages. The macrophage engulfs the antigen and sits it in a vacuole. Lysosomes then like to join the party at this point by inserting digestive enzymes such as enzyme lysozyme into the antigen, effectively destroying the antigen and breaking it into harmless smaller particles. The macrophage then most often releases these harmless smaller particles. However, they do keep some of the particles from the antigen to present on their major histocompatibility complex 1 and 2. This part is for the third line of defense. I haven't lost anyone, have I? Good, let's move on. Now the link between the two processes is the movement of the antigen-presenting macrophage to the lymph nodes. After it's moved, it will come across a matching helper T-cell which will recognize the macrophage is presenting an antigen. The matching helper T-cell will then bind to the MHC class 2 antigenic peptide complex, or just simply MHC2, and become activated, after which it releases chemical messages such as cytokines. Here is our crossing point into the third line. The cytokines and other chemicals activate and stimulate corresponding T-cells to clone many copies that are specific to the antigen. Some will become memory T-cells, which combine with the MHC2 of a macrophage in order to obtain the protein and keep a record of it in case the body is infiltrated by it again. Then they remain active for a while, just in case the same infection were to happen again. Most T-cells, however, will become cytotoxic T-cells. This is where I come in. After the memory T-cell has obtained the antigen's protein, I will usually dock with the MHC1. The MHC1 are responsible for alerting the killer T-cells. They themselves need to be destroyed. All nucleated cells have the capacity to destroy antigens which infect them, but they also have MHC1 complexes. If I sense that a cell has presented an antigen protein on the surface of their MHC1, I need to bind with them and release a potent chemical called perforin. It's all fun and games until it comes to that point. Anyway, thank you for watching.